Hi everybody, I'm Emanuele and I'm excited to present you today an entry point introduction to the RNA world. Here you can find my Unime email address if you want to contact me for more in-depth knowledge about this topic, both from computational and biological sides. To better understand the RNA world, we'll have some background knowledge, background information about cellular and molecular biology. We will follow a top-down approach, so we start from the macro, so the cell, going to the micro, so nucleic acids, focusing onto especially RNA, which is the main focus of this presentation. So let's now start talking about what is a cell and explaining the main differences between eukaryotic and prokaryotic cells. The cell theory states that the cell is the biological unit of all living beings. And organisms can be divided into unicellular, so organisms made up of only one cell, and multicellular organisms that are made up of a variable number of cells depending on the species we are considering. All cells share a common biochemistry, so they are all made of carbohydrates, proteins, nucleic acids, minerals, lipids, that are fats, and vitamins. And this is very important for the purposes of this presentation, that every form of life uses DNA as a genetic material. Cells can be further divided into prokaryotic and eukaryotic cells. So prokaryotic cells are simpler and older, and by older I mean that they have been in existence longer on the Earth than eukaryotic cells that can be thought as an evolution of prokaryotic cells. What is important for the purposes of this presentation is that the prokaryotic DNA is not surrounded by a nuclear membrane, but a nucleoid region, a not well-defined nucleoid region, is still present. Instead, in eukaryotic cells, the cytoplasm contained a well-defined nucleus in which the DNA molecules store the genetic information. And this nucleus is enclosed by a nuclear envelope, so we have a well-defined region in which genetic material is present, and also various organelles are present inside the cytoplasm that execute the vital functions for the cell. Let's now examine the principal cellular organelles of the eukaryotic cell. All these organelles are found inside a cytoplasm, which is enclosed by a cellular membrane, allowing ordered organization in and keeping chaos outside the cell. Inside the cytoplasm, we have these organelles and also the nucleus, in which genetic material is stored as DNA molecules. Within the cell, biomolecules building and dismantling reactions happen. And to achieve this purpose, we need energy. And the first organelle we are going to see is the mitochondrion. The mitochondrion can be seen as the site of ATP synthesis. A series of biochemical reactions known as Krebs cycle occurs here. And starting from glucose, so basically a sugar, we obtain ATP, which is a fundamental molecule in which energy is stored. The plant counterpart is called the chloroplast, and the chloroplast is the site in which, starting from carbon dioxide, water, and photons, so basically sunlight, we obtain glucose and the oxygen we all breathe. Another important organelle is the endoplasmatic reticulum, which can be further subdivided into rough endoplasmatic reticulum and smooth one. The rough one is rich in ribosomes. We will discuss in more detail later ribosomes when talking about what is a protein and protein synthesis. The smooth one instead is devoted to phospholipids synthesis. Phospholipids are a key component of cell membrane. The Golgi apparatus instead can be seen as a dispatch station of protein products from the AR, delivering them to their final destination. Lysosomes are the recycling stations of the cell, and degradation, so catabolism, means energy as a discard, so lysosomes contribute to cellular homeostasis. All these organelles are not moving freely inside the cytoplasm, but a cytoskeleton, for they are subdivided into microtubules, intermediate filaments, and microfilaments, keep them all anchored. So let's now start talking about DNA molecules that carry genetic information essential for cell survival and cell reproduction, focusing onto DNA structure and purposes. DNA molecules are long polymeric chains of small subunits called nucleotides. Nucleotides are made up of three key elements: deoxyribose, so a pentose sugar. You can see its structure with five extremities here. A nitrogenous base, so called because contains a nitrogen atom, and a phosphate group containing phosphorus instead. There are only four possible nitrogenous bases and therefore nucleotides inside DNA, and adenine and guanine have a bicyclic chemical structure, and therefore they belong to purine's nucleotide category, while cytosine and thymine have only one ring and belong to pyrimidine category. DNA has a structure with high stability and in vivo, so if temperature, acidity, so pH, and humidity are the ones characteristic of living cells, it is always found in the double helix form, so two strands that wrap around a common axis. This structure was discovered by Watson and Crick, helped by Rosalind and Wilkins' X-ray crystallography studies. Nucleotide bases are arranged in the internal part of the double helix, and nucleotides pairing follows a strict pattern. Adenine can bind only thymine, and cytosine can bind only guanine. The backbone sugar phosphate is the external part and is kept together by phosphodiester bonds that are strong chemical bonds, while nucleotides inside are bound by H bonds, which are easier to break. This is very important because it allows DNA to be replicated in an easy way by breaking H bonds and copying one of the two strands. DNA contains all the genetic information that is necessary for the life of the organism, and we can say that DNA is the same in every cell, bar few exceptions, so that are called mutations that can occur inside during DNA replication. Eukaryote, then human DNA, is found inside the nucleus, as we have already said, and the cellular nucleus holds many nuclear pores that regulates import and export of biomolecules. At this level, segments of DNA can be called genes. And we have around 2 meters of DNA per cell, so DNA molecules need to be packed. And nucleosomes are composed by DNA plus a specific type of proteins called histones. Linked nucleosomes form chromatin, which can be divided into open and closed chromatin. Chromatin is hierarchically coiled and supercoiled with these beads on a string fashion. And without going into further details, you can imagine that having open or closed chromatin, so packed or less packed chromatin, and so DNA topology with less and more packed DNA affects DNA accessibility. DNA and therefore chromatin gets organized in structure called chromosomes, with prokaryotes that have only one circular chromosome, while eukaryotes have N chromosomes, with N defined by the species we are considering. I don't want to go into further details about DNA complex organization, which would be outside of the scope of this presentation, but if you are really interested, I can for sure provide you some material by email. This slide is just to show you the 23 homologous chromosomes in human somatic cells. Specifically, here we have a male because we can see this XY couple that in female individuals is substituted by an XX couple. We have just talked about DNA, so that now we can focus on to RNA molecules. From a structural point of view, RNA is a polymer of nucleotide, basically the same as DNA, but with some differences we are going to explain now. So we have ribose as sugar instead of deoxyribose, and ribose is a less stable sugar than deoxyribose. Then we have 3 out of 4 nitrogenous bases that are the same as DNA, so we always have A, C, and G, adenine, cytosine, and guanine, but instead of thymine, as you can see also in the picture below, we have another nitrogenous base that is very similar, but is called uracil. So ACGU and U instead of T, that always matches with adenine A. RNA is always exists as a single chain, and at this level of the presentation we haven't introduced yet transcription, but you can imagine that this single chain composed of which RNA is composed by is synthesized starting from one of the two chain that compose the double helix of DNA molecules. And these bases composing RNA interact in complex three-dimensional structures. We'll see in the second part of this presentation something about that. And this is a new field of research, and these structures often are not well known yet. As I already briefly introduced, RNA molecules starting from one of the two synthesized starting from one of the two chain of DNA directs the protein synthesis. So we start from DNA, then RNA, and then proteins. We haven't introduced proteins yet, so we can now introduce proteins that are polymers of amino acids. To make a comparison, we have just said that DNA and RNA molecules, so in general nucleic acids, are polymers of nucleotides. This does not occur by chance. We will see that starting from a DNA molecule, so a polymer of nucleotides, an RNA molecule is synthesized. So always a polymer of nucleotides, but arranged in a single-chained fashion. And RNA carries the information needed to generate a protein. Always a polymer, but a polymer of amino acids. And linked amino acids form proteins, and changing the order of amino acids, we have different proteins, as changing the DNA nucleotides leads to different genetic information. Amino acids are biological entities, biochemical entities composed by one central atom of carbon, bonded with one atom of hydrogen, and one amine group that contains a nitrogen atom and a carboxylic acid that contains two oxygen atoms. What is very important to remark here is that we have also a side chain. We have 20 possible side chains that leads to 20 different proteins, amino acids that bonded together form proteins. And I want to remark that we have a common structure as nucleotides, but different side chain that gives rise to 20 different proteins, such as four possible nitrogenous bases leads to four different nucleotides. Proteins can be seen as the effectors of all functions within the cell, and here I just reported some of them to give an example. For example, energetic, immune, structural, transport, enzymatic, hormonal, signaling, and contractile function. Contractile means what happens in our muscles that are composed by fibers of proteins and allows our movement. So, having introduced DNA structure, RNA structure, and the final outcome, so proteins, we can now start talking about RNA that codifies and codes proteins, different types of proteins, which is called coding RNA. To introduce coding RNAs, we need to understand better the process by which a protein is synthesized, which is stated by the central dogma of molecular biology, that defines how we transfer the genetic information from DNA to RNA to proteins. This occurs in a one-way fashion, even if some exceptions exist. We know that in science there are no dogmas. And starting from DNA, we obtain RNA molecules in a process called transcription. Then, from the RNA, the just obtained RNA molecules, we obtain the protein by another process called translation. This is very important to keep in mind. And proteins are not the final outcomes of this process. In fact, proteins need to undergo post-translational modifications to really have the metabolic stability and to operate in a functional way. The main actors of this process are DNA, we already talked about it, and proteins, we talked about it, and different types of RNA, in particular messenger RNA, ribosomal RNA, and tRNA, transfer RNA. If you remember, at the very beginning of this presentation, we introduced different cellular organelles, in particular this rough endoplasmatic reticulum rich in ribosomes. And ribosomes are the machinery that allow us to transfer information from RNA to proteins, and ribosomes are constituted by RNA and rRNA, so ribosomal RNA, and specific types of proteins. This is what happens at a molecular level. We have DNA carrying genetic information with its double helix structure transcribed into messenger RNA. We can see that one of the two helix of DNA is transcribed into mRNA, and that bases are complementary. So if we have a cytosine in the DNA molecule, we'll have a guanine in the mRNA, and vice versa. The only difference is that in mRNA we have uracil instead of thymine, but uracil always pairs with adenine. It's also very interesting to note that following this pattern, the mRNA molecule is a copy of the other helix of DNA that, if you remember, carries the same genetic information but in a reverse complementary way. Then mRNA exits the nucleus through nuclear pores and undergoes chemical modifications that increases its stability. The mature mRNA molecule is therefore scanned by the ribosome machinery here depicted with its two subunits in which mRNA goes by. The final ingredient we need to move from nucleotides to amino acids is transfer RNA. tRNA that acts as a linker between triplets of nucleotides inside the mRNA molecules called codons and amino acids. It has this cloverleaf structure carrying an anticodon complementary to the mRNA molecule, the mRNA codon, and on the other side, the specific amino acid encoded by this codon of mRNA molecule. Finally, amino acids can be linked together with peptide bonds to form the final protein. So we started from DNA and obtained mRNA in a process called transcription. Then from mRNA we obtain messenger RNA, we obtain a protein in a process called translation. Let us now spend a few more words about messenger RNA. We have just said that messenger RNA corresponds to the genetic sequence of a gene. We said a gene is a segment of DNA, and this process is called transcription for DNA, and then messenger RNA is read by ribosomes in the process of synthesizing a protein called translation. In eukaryote, another mechanism that is important to mention, also because we will encounter this mechanism in the non-coding RNA section of this presentation, is called RNA splicing. Splicing is a mechanism by which introns that are non-coding regions, here depicted with lines of the non-mature mRNA, are removed, excised, and exons, that are these rectangles, exons, that are coding regions, are joined. We also have this particular process called alternative splicing, that in which particular exons of a gene may be included within or excluded from the final processed mRNA. So we can have a different combination of exons joined together in the final mature mRNA molecule. So having different combinations and therefore different mRNA strands, we have different codons and so different translated proteins. And often this is reflected by different biological functions. The group of rules defining how the information stored inside the nucleotide sequence in mRNA molecules is translated into amino acids that compose proteins is defined as genetic code. So we need to start from a 4-basis alphabet, so an alphabet composed by 4 letters, so adenine, guanine, cytosine, and uracil. Uracil, I remark that is the equivalent of thymine in the RNA molecules. And we need to start from a 4-basis alphabet and go to 20 amino acids alphabet, 20 amino acids that constitute with different orders, different proteins. So this code has two remarkable properties. The first one is that it is indeed universal in the sense that it is valid for most all organisms. The second one is more related to something more close to information theory, so to a computer science field, and it is the redundancy property. So in mRNA codons, and I want to remark that codons are simply sequences of 3 nucleotides, one after the other, inside mRNA molecules. we have a error correcting code because we need to start from a 4 basis alphabet, so an alphabet of 4 letters, and we need to go to a 20 amino acids, 20 letter alphabet. So the minimum number of transformation we need to achieve this task is to raise to the power of 3 the 4 basis alphabet we start from. So doing this mathematical type of operation, this arithmetical type of operation, we achieve 64 possible codons for 64 possible codons that encodes for different amino acids. But doing this, we can have in a mathematical way a bijective function, I'd say, because we start from 3 different nucleotides sequences that encode for the same amino acid. Just to give to show you an example is for so proline. We can generate proline using the type of machinery we considered before, talking about the central dogma of molecular biology and RNA information passing onto amino acid biomolecules. So we start from different type of codons. For example, here we have CCU, CCC, CCA, and CCG that encode for the same type of amino acid. So we have this type of redundancy that is reflected in an error corrected code. In fact, if we would have a mutation inside this starting point, starting codons, we still have this amino acid that is generated. Of course, here I proposed just a brief summary of what happens at the molecular level in the transition from DNA to RNA to protein. We also have other types of ingredients. We have, for example, transcription factors that are particular types of proteins that stimulates or repress the transcriptional apparatus. And I want to remark that we have the same DNA in every cell of our body except some mutation that can occur. But we have different gene expression patterns that give rise to all our different tissues and different organs in our body. This transcriptional complex apparatus program is determining the most of the cell diversity, and this regulation can be achieved with transcription factors, but also by non-coding RNAs, which is the main focus of the next section. I just want to mention 2 types of small non-coding RNA that we are going to see better in the second part of this presentation, and they are small nuclear and small nuclear RNAs that primarily guide chemical modifications of RNAs, mainly ribosomal RNAs, tRNAs, transfer RNAs, and themselves. So also different types of drugs have been proposed that and they work at this level of regulation to restore physiological states from pathological ones. So here we are with the second section of this presentation about the RNA world. This second part deals with non-coding RNA world, a new field of research that focuses on 2 RNA molecules that do not encode proteins. So as we said, functional RNA molecules that are not translated into proteins are called non-coding RNAs. 97, 98% of our genome is non-coding, and genome in general is the genetic material of an organism, so the DNA contained in each cell of an organism. And prokaryotic genomes are almost totally composed by protein encoding genes, while, as I said, eukaryotes have low gene density. So 97, 98% of eukaryote genomes are non-coding. And if you consider that the 30% of coding part is devoted to DNA maintenance and integrity, this percentage drops to about 0.8%. And similarly, the transcriptome can be defined as the set of all RNA transcript in an individual. By explaining the mechanisms of action and regulation of these types of RNA, we want to debunk the notion that this type of RNA is just junk, as it has long been theorized and nicknamed. The first type of non-coding RNA we are going to see are long non-coding RNAs. And long non-coding RNAs are basically RNA transcripts with more than 200 nucleotides. So this arbitrary limit helps us to distinguish between long and small non-coding RNAs, small non-coding RNAs that we will see later. The long non-coding RNAs are the majority of transcription products, and they seem to recruit and guide the protein involved in modifying a chromatin structure. And I remember you that chromatin is the method by which DNA is stored inside the nucleus of our cells. And long non-coding RNAs seem to have a primary role in disease, especially neurological disease and cancer. And using small non-coding RNAs strategies such as antisense oligonucleotide and short interference RNA, we will talk about them later. And they seem to target long non-coding RNAs and to have an effect on tumor initiation, progression, or metastasis. A particular type of long non-coding RNAs are circular RNAs, and they are generated by splicing non-canonical splicing events. On the left side of this picture, we can see the already mentioned canonical linear splicing. We talked about it before, and that leads to linear mRNA. On the right side, we can see non-sequential backsplicing of exons, these colored arrows, and introns or combinations of both, as this one. And a brilliant paper that explains very well the mechanism at the basis of these non-canonical splicing events is this one, and I provide you the link in case you want to go into further details. These circular RNAs are differentially expressed, so are present in different quantities with respect to non-pathological cells. They strongly seem to interfere with small non-coding RNA functions. So, having mentioned small non-coding RNA, we can go into further details and say that small non-coding RNAs in general are transcripts with less than 200 nucleotides. And obviously, being non-coding RNAs, they are not translated into proteins. Small non-coding RNAs are involved in several cellular biological pathways, including translation processes. We already talked about it in details. The process by which we start from RNA molecules and obtain proteins. Splicing mechanisms, so we talked about them, and particularly alternative splicing. Also catalysis, we will see something about it when it comes to ribozymes. And catalysis in general is mechanisms that accelerate biochemical reaction that normally occur inside our cell, our body. Also gene editing, we will talk more in detail about it when it comes to guide RNA, so directly related to CRISPR-Cas9 technique. And the last biological pathway they are included is RNA interference, RNAi pathways. And I want to spend some few words about it because it's at the very basis and very useful to understand the mechanisms of action of different small non-coding RNAs. This is an example of RNAi, so RNA interference pathway. We have our small non-coding RNA molecule strand that is able to recruit the RISC, so RNA-induced silencing complex. And once inside the RISC, the small non-coding RNA pairs, complementary pairs, with a target mRNA molecules that has already exited the nucleus throughout nuclear pores. And the small non-coding RNA so pairs complementarily with mRNA before mRNA is converted into a protein by a ribosome. If you remember, the ribosomes are the machinery by which we transform we produce proteins starting from molecules of RNA. And this complementary basis pairing system is the same as what occurs in DNA. So A pairs with U in this case because we are talking about RNA, so we are dealing with uracil instead of thymine, but the mechanism of the basis is the same. So adenine pairs with U. So for example, if this is the small non-coding RNA strand, the adenine inside of it pairs with the uracil inside the mRNA, the messenger RNA target strand. And so by complementary pairing inside the RISC, they are kept together. And the RISC complex is therefore formed by this small non-coding RNA paired with the target RNA, but also has a catalytic property, catalytic in the sense of cleavage. In fact, RISC is also nicknamed as molecular scissors because the RISC complex has a cleavage catalytic site, and inside of it the target mRNA can be cleaved, and this target mRNA can belong to any target, to any gene inside our genome, and therefore it can be cleaved and then degraded. I provided also a link to a YouTube video in which this pathway, this biological pathways, is well explained. So now that we have a better understanding of small non-coding RNA mechanisms of action, we can go into further details and talk about different types of small non-coding RNA molecules. Particularly, we already talked about ribosomal RNA, transfer RNA, small nuclear and small nuclear RNA that can be clustered into translation-related small non-coding RNAs. So we will focus on other types, important types of small non-coding RNAs. And we start looking at microRNAs. MicroRNAs are a particular subtype of small non-coding RNAs that block translation or promote the degradation of messenger RNAs by RNA interference mechanism we discussed before throughout complementary base pairing. And what is very interesting, the peculiarity about microRNAs is that they do not require a perfect base pairing, and so they can have multiple target messenger RNA molecules. Another expression of microRNAs or oncogenic microRNAs is present in tumor tissues and cells, so higher quantity with respect to physiological state cells, and a technique called Myrna mimics using synthetic microRNAs, so studied in and produced in vitro in lab, is under current evaluation. So some tumor suppressive mRNAs lost in cancer cell may be restored by using this synthetic Myrna mimics. And what is very interesting about microRNAs and the developing of microRNA-based drugs is that the microRNAs and the RISC complex, they are based and they are inside are endogenous components inside the cell, and so Myrna microRNA drugs may be well tolerable in cells. But we haven't FDA approved Myrna drugs as of today because their chemical modification isn't easy to do, and also because of this peculiarity. We don't want to have multiple target when it comes to specific and synthetic new Myrna-based drugs. We want to target one single mRNA molecule and do not interfere with others that also may be important, may be vital for the cell living. siRNAs, short interfering RNAs, act similarly to microRNAs by blocking translation or promoting degradation of specific target messenger RNAs. And the difference between siRNA and Myrna is that siRNAs can promote the degradation of specific target mRNAs. So they tend to have only one single target mRNA molecule. Therefore, having a specific action on a specific biological pathway. And chemical modification is a common strategy to improve metabolic stability of in vitro siRNAs and to improve pharmacokinetics properties. There are 3 FDA-approved siRNA drugs, and the peculiarity of siRNA, beside the fact that they are easily modifiable, is that they are endogenous, and the RISC complex they are part of is endogenous to eukaryotic cells, and thus does not require the delivery of, for example, large enzymes with specific protein domains. And finally, given that siRNA interferes with mature mRNA, it requires only cytoplasmic delivery, which is easier to achieve than nuclear delivery. Antisense oligonucleotides are complementary sequences of target messenger RNA, and they act always by using RNAi mechanism. They have three main mechanisms of action, and they can interact with pre-mRNA, so non-mature mRNA, and cause alternative splicing. We talked about it in the first part of this presentation. They can also cause translational arrest or also RNA degradation. RNA degradation that occurs not by recruiting the RISC enzyme we talked about before, but with another enzyme, non-specific enzyme called RNAH1 in this case. And another interesting type of ASO are called gapmers, and gapmers are sequences of RNA and DNA hybrid. So we have RNA mimics at the beginning and at the end of this gapmer strand, and we have the DNA between them. And this hybrid RNA DNA hybrid can always, with the same mechanism as before, so RNA interference, pair with target mRNA molecules, and then they can recruit the RNA H1, for example, and promote and RNase H promote the cleavage and the degradation of mRNA molecules, and we also obtain nucleotide we can recycle. A difference I want to show you between siRNA and ASO is that siRNA are more effective to knock down cytoplasmic target, because the RISC, the complex RISC, is more present in the cytoplasms of the cells. Instead, RNase H is more abundant highly abundant in the nucleus, and so ASOs are more effective to knock down nuclear targets. So back to us. ASO are also very important genome editing tool, in this case RNA, so transcriptome editing tool. And because they can recruit the endogenous RNA editing enzyme, ADAR. And this enzyme, in this figure, we can see a very similar structure with respect to the RISC complex we talked about, inside which we have the target RNA, mRNA messenger RNA, and the ASO pairing, complementary pairing. And but instead of differently from RNase H1, for example, and RISC complex, this ADAR complex can act as an editing tool, RNA editing tool, converting the nitrogenous base A into I. We haven't talked about inosine, but we can thought of it as a very similar nitrogenous base to G. In fact, we pass from a target RNA with base A to a modified RNA with base I, which is read as G by the translational machinery, and therefore it can be read as a guanine instead of an adenine, as in the original molecule. Chemical modifications are always easy to achieve and improve metabolic stability of these oligonucleotides, and they are essential to reduce cytotoxicity and cell penetration efficiency. 3 FDA-approved ASO drugs exist today. The next subtype of small non-coding RNA are aptamers. Aptamers are short because they are small non-coding RNAs, single-stranded nucleic acids, so RNA, that bind to a variety of different targets, such as protein, carbohydrates, DNA, so RNA. And what is important to know about aptamers is that they act by virtue of their tertiary structure rather than their sequence, as in the types of RNA we consider, the 3 types of small non-coding RNAs we already considered. In fact, this is the simple sequence of nucleic acids, one after the other. And aptamers do not act by RNA complementary base pairing, so at a sequence level, but as they act at a 3D, three-dimensional structure level. So forming this peculiar 3D structure, they can bind to a market and target it. And they are also well studied because they can be carriers for other drugs because of this 3D structure. And they bind to proteins that are traditionally targeted by small molecule drugs, potentially replacing them, because at the end of the day we remember that aptamers are RNAs and therefore nucleotidic sequences. And nucleotidic sequences are easy to build in vitro and easier to build than small molecule drugs. As of today, we have only one FDA-approved drug because we have an issue with aptamers is that they are sensitive to degradation by nucleases. Nucleases are basically enzymes, so proteins that cut nucleic acids and particularly RNAs. And but this issue can be solved by the means of possible chemical modifications, as we have already seen with sirna and ASOs. Riboswitch have emerged as potential therapeutic targets because they are highly structured to form a unique ligand binding pockets and function as gene regulatory factors. Here you can see this peculiar stem loop structure in which they can contain, for example, mRNA and may provoke alternative splicing and self-cleavage to cause mRNA degradation. They are commonly found in bacteria, but also discovered in other biological domains such as plants and fungi, and they directly control gene expression critical for survival, especially in the bacteria domain. Here I also provide you a link to a paper in which this particular structure is illustrated. We then proceed from riboswitches to ribozymes. We always have this prefix ribo because we need to remember that RNA is the shortening for ribonucleic acid. In fact, at the biochemical level, we have seen that we have a sugar, ribose. And ribozyme is a word, crasis, for ribonucleic enzyme. So they are RNAs, small non-coding RNAs, having enzymatic action. So they catalyze, they speed up biochemical reaction. For example, mRNA and protein cleavage, but also other types of biochemical reactions such as esterification and also hydrolysis of phosphodiester bonds. If you remember, phosphodiester bonds are at the basis of the sugar phosphate backbone of DNA. Here, for example, you have the hammerhead ribozyme, and that targets a particular type of mRNA molecule. It forms a complex, always with by complementary base pairing. But what is very interesting is the fact that this type of small non-coding RNA has an enzymatic activity itself. So without requiring any RISC or RNA-H complex, ribozymes can achieve mRNA cleavage. And because of it, synthetic ribozyme directed against HIV RNA virus has entered in the clinical testing. Similarly, ribozyme have been designed to target other types of diseases based on retrovirus and more in general virus based on RNA. Also, for example, SARS-CoV-2. The very last category of small non-coding RNA I want to mention is guide RNA. And we can't talk about guide RNA without mentioning CRISPR-Cas9 technique, which led to the Nobel Prize in Chemistry in 2020. Guide RNA is a very interesting type of non-coding RNA because it, as its name says, guide the CRISPR-Cas9, which is a nuclease, so an enzyme, a protein that cuts nucleic acid towards a target gene. And this is very important because we are guide RNA and Cas9 can act at an RNA level, but more interestingly at a DNA level. So we can precisely edit genome sequence toward irreversible knockout or knock-in of a target gene, for example, in mammalian cells and organisms, as compared with RNA interference, which does not completely eradicate gene expression because it acts at a transcriptome level, so working with RNA transcript and not DNA, which stores the genetic information. Here I wanted to provide you an RNA classification that can help us while if dealing with new types of RNA molecules. So starting at the top of this tree, we have RNA node, and we always have to ask us if this new type of RNA encodes a protein. If the answer is yes, then we are dealing with a coding RNA, so which can be only of one single type, so messenger RNA. That we saw it's related to translation-related RNAs such as R-ribosomal RNA, so involved in ribosome, and small nuclear, and transfer RNA, which is the linker between amino acids and codons stored in mRNA molecules. If the answer then is no instead, we are dealing with non-coding RNA. We have a non-coding RNA which can be further subdivided into long non-coding RNA if we are dealing with RNA with length, a physical, not a logical, a physical length of more than 200 nucleotides. Instead, if we are dealing with RNA with less than 200 nucleotides, we are in front of a small non-coding RNA. And we have just seen that small non-coding RNAs can be divided in many sub-branches. We, for example, have just seen ribozymes, miRNA, and ASO, aptamers, or riboswitches, but many other exist. And if you want to go into further details, do not hesitate to contact me. The last section of this presentation is related to RNA drugs discovery and therapies. We start talking about RNA drugs and therapies, recapping the main mechanisms of action we have already seen. We have seen that RNAs, particularly small non-coding RNAs, can manipulate gene expression by RNAi mechanism. So, for example, RISC complex or RNase H, or also by the means of their tertiary structures, such as in aptamers. Another mechanism is mediating DNA or RNA editing. We saw it when talking about guide RNA directly related to CRISPR-Cas9 technique. Another mechanism of action is the encoding of protein, and this is called mRNA therapy. We will see at the end of this slide something more about it. And I want always to remember you that from RNA we pass on to protein by the so-called process of translation. Another mechanism is to targeting RNAs or proteins. For example, we saw it when it comes to hammerhead ribosome. And all these mechanisms of action are really interesting for pathological states and, for example, human cancer, but more in general, human diseases and genetic disorders. Here I want to also mention an example of RNA drugs and RNA therapy, which is the mRNA vaccine discovered used against the SARS-CoV-2 virus. And this belongs to this category of mechanism of action. In fact, this mRNA vaccine carries an mRNA molecule inside a liposome, so a circular lipid, a circular fat, that can easily enter the cell. And this is a well understood now mechanism by which we can transfect exogenous, so built in vitro messenger RNAs into mammalian or, for example, human cells. So we have our mRNA that encodes for a spike protein. The spike protein is present on top of the SARS-CoV-2 virus. If we want to go into further details, this is present in the capsid, the proteic envelope that contains the viral information, the virus itself, so the RNA virus. And by using the ribosomal machinery, we can scan our mRNA and translate it into the spike protein itself. So this spike protein can be targeted by our immune system, by our antibodies, and when the virus, the SARS-CoV-2 virus really occurs, we already have antibodies against its spike protein. So we just consider different mechanisms of action of small non-coding RNAs, and we saw a particular implementation of them, particularly mRNA vaccines. And starting from those mechanisms of action and from systematic classification we can find in literature, for example, this one, which divides small non-coding RNA into different categories according to their mechanism of action, for example, RNA interference that can promote degradation and inhibit translation. We start from these considerations and we amplified the pre-existing differentiation between RNAs as medications and as targets. So, for example, we have RNA as medication, for example, ASO or siRNA, and then RNA that can be targeted by other RNA molecules, but also by other completely different types of small molecules or drugs. Here I want to show, for example, what happens in a well understood mechanism that deals with and targets our ribosomal RNA. I want to remember you that ribosomal RNA is part of the ribosome complex with other types of specific proteins. And in this section view of the ribosome, we can see that specific types of antibiotics, so we are not talking at all about other types of RNAs, but we are talking about antibiotics that particularly and specifically a macrolide. Macrolide are microcycles of esters, and you can see here that this particular type of macrolide with this very complex structure can block the peptide that is the protein that is exiting the ribosome, and therefore blocking and stopping translation process. So here in this slide we recap advantages and challenges for RNA drugs. Advantages, basically they can target undruggable pathways or for small molecules or proteins. They are rapid and cost effective to develop. In fact, we always have to consider, as we already did when we talked about aptamers, that RNAs are basically sequences of nucleotides, and they are very cheap to build in vitro in a lab. Also, this leads to a very important consideration, that is they can rapidly adapt to the variation of new viral forms, new evolving pathogens, such as what is occurred with SARS-CoV-2 new variants. Also, because we are dealing with RNAs, we have no risk of chromosomal integration that can occur instead when it comes to DNA drugs. If you remember, the DNA is organized in a chromosome in our genome, the DNA inside all our cells, and so DNA drugs have the risk of integrating into our chromosomes. Challenges: RNA can be obstacled by RNases, that are specific enzymes, specific proteins that cleave RNA molecules, and this is an obstacle for delivering, for ferrying RNA to the desired location. Another challenge is to overcome the cytoplasmatic barrier, and this is brilliantly done by RNA vaccines, as we briefly mentioned before, by using liposomes, so lipids that contain the mRNA molecules. They are not easily oral bioavailable, and another challenge is, as we previously have seen when talking about microRNAs, is the selectivity toward the target and the toxicity at a cyto, so at a cellular level, cytotoxicity. This last slide is intended to show you the work we are currently involved in, and basically I want to show you that all the notes of this metagraph we are developing that contains different types of RNAs, particularly non-coding RNAs, and basically the ones we consider during this presentation. And I wanted to show you how it is important to know the biological meaning beside different RNA entities, even if we are building a knowledge graph that is computer science matter. So these are the references for this seminar, and feel free to contact me if you have any doubts or curiosities.